The goal of this experiment is to have students test milk products and milk for the presence of various biomolecules. This is kind of an investigation of milk and milk products that students may be used to seeing on the shelves in the grocery store or in their homes. So the data sheet will walk them through a few questions just to get the juices flowing to help them formulate their hypothesis. One of the things that we are working on, one of the skills that we're working on this semester with students is developing a hypothesis based on pre-existing knowledge. So thinking about what they already know and making an educated guess and then formulating a hypothesis that they will test using these three reagents. So students should spend some time working on this part. And then we also want them to think about the concept of a positive control and a negative control. So for each of the tests, we want them to write down what their positive control and negative control will be. And this is kind of outlined in the PowerPoint slides. Next, the students will perform the BioRet test to test for the presence of protein in their products. So students will take a photo of their products before, and they'll have their ceramic plate. And then we want them to use this space here to make some observations as to the color. Is it clear? Is it opaque? And then here we want them to discuss or just describe like one or two words for a color change. And the point here, the whole purpose of these images and having students write down something is we want them to make a decision with their group members based on what they're observing from their experimental results. So we want like a simple yes or no, or a check mark or something kind of right here in this space. And we want them to write it down so that when they come back to their conclusions, they can say, you know, did we observe a color change? Did we make a definitive decision as to whether protein is present or not in our sample? And then they can discuss their results. We have our beakers. Each lab room will have beakers with milk and milk products labeled 1 through 6. And then instead of students kind of running around the lab and grabbing milk from one place, each table will come and get their working solution of milk and milk products and bring it back to their table in test tubes. And we figured that about 3 mLs, this doesn't have to be exact, they can measure with the bulb pipettes here. They can measure about 3 mLs of each solution and bring it back to their table to perform their 3 tests. So this will kind of be their working solution to do each of the 3 tests.
All right, students will measure 200 microliters of the appropriate milk product into each well of the spot plate. Different milk products have slightly different colors, so we want students to recognize this. Finally, number 6. Oops, I'm gonna throw it away too early. There we go. All right, so we have a positive control, which is going to be egg albumin solution. Have this in a beaker as well for the students to use. I don't have a beaker with it in it right here, but we'll put that in here. And negative control is going to be water. So let's go ahead and put that into the negative control well. Okay, at this point, students should take a photograph of their spot plate.
Carry on.
All right. Students will then add approximately 250 microliters of BioRet reagent, and we have it in these dropper bottles. So 250 microliters is approximately 5 drops. So let's go ahead. There's 1 drop, 2, 3, 4, 5. In that one so far. Okay, just keep doing this. Three, 4, 5. Okay, now we need to mix the plate a little bit. Let's see if this works. Okay, when we take a look at the final colors, we're going to recognize that there's going to be gradation all the way from blue to purple. So in some of these we see a lot of purple, some of these we see just a little bit of purple, and some of these are fairly blue still, but maybe a little tiny bit of purple. So that's different amounts of protein in the milk products. So each table will have a waste beaker for the BioRet waste. And what we're going to have students do is use a bowl pipette to basically remove as much of the liquid as they can from each well of the spot plate and put that into the BioRet waste beaker.
For the Benedict's test, students will take 1 ml or 1,000 microliters of each of their samples. So you can see here I'm working with samples 1 through 4 and place 1 ml of each sample into their test tube. And they should be changing out pipette tips so as not to cross-contaminate between different milk products. For the positive control, we'll be using a glucose solution. And for the negative control, we'll be using water. Then they'll add 1 ml or 1,000 microliters to each tube of Benedict's reagent. And here they don't have to change the pipette tip since we're not submerging the pipette tip into the solution. So 1 ml of Benedict's reagent into each of the tubes. Okay, next, as you can see, all of the solutions are still either deep blue or cloudy blue color. They will place the test tubes into a boiling water bath for 2 minutes, and 2 minutes is more than enough time to see a color change. Please be careful not to drop the test tubes in the water bath, just to gently place them. We're using a 250 ml beaker here, so it's small enough. After 2 minutes, we see that our solutions have changed color. And here are the results. So the test tube 1 and 2 have an orange color, demonstrating the presence of reducing sugars. Test tube 3 is negative. Test tube 4 is like a greenish purplish, which indicates a very small amount of reducing sugar. Test tube 5 was our positive control and it's a deep red, and then test tube 6 was our negative control and it did not change color. The Benedict's reagent is hazardous and needs to be disposed of properly. Students should dispose of the reagent into the waste beaker. Beakers at their tables, and then photographs of their test tubes before and after. Here they can fill in their observations of the color before and the color after. The last test is the iodine test for the presence of starch. So students will pipette 500 microliters of each milk product into the plate. Sample 2 goes in well 2. Sample 3 was a weird one. Sample 6. And we have, again, for our negative control, water. And for our positive control, we have a starch indicator solution. Right there. And then students will add the iodine solution. 1, 2, 3, 4, 5 drops in each well. You can see the starch solution has a drastic change in color. And then you can use a toothpick to mix the solutions. And use the other side. Okay, we've got some color change there. And no color change there. So as you can see from these results, the product number 5 is the only one that had a reaction for the starch solution, for the starch test. Okay, we might have some varying results depending on what type of oat milk is used for sample number 3. Sample number 3 here showed a negative test for starch, whereas when I used a different brand of oat milk, it showed a positive test for starch. I'm not sure what's going on with this brand of oat milk. I think maybe it is sweetened oat milk and the sugars in the solution maybe are not providing an environment for the starch and the iodine to bind, as iodine binds to those coiled up polysaccharides. So here we have what the oat milk or sample 3 should look like. And we may have varying results on the different campuses. So samples 1 through 4 showed negative results. This should be a positive result. This should be this right here. Sample number 5, little bit of starch, and sample number 6, no starch, whereas our positive and negative controls showed the exact results that we would have expected. The negative control stays this amber color, the water, and then the starch solution turns this deep blue-black solution, just as we've kind of seen in this additional solution, this additional test that I did with the Kirkland oat milk, so unsweetened oat milk. For the iodine test for starch, students will do something similar where they will record their observations and take a photo of their ceramic well plate before and after adding iodine. Once they've added iodine, they can make observations and make a definitive decision as to whether or not they think starch is present. And then they will go and look at their results for each test to decide whether or not each test had protein samples present, which is why it's really important for them to take good observations and write down what they think they see in each test Once they've done this, once they've spent some time doing their analysis and conclusion, they can look at the list of substances or the list of milk and milk products, we should have 4, that they have in their lab manual, and we will present it to them on the slides, and they can make a decision as to what they think it is, and then we will let them know what it actually is on the slides.